Malaria is a tropical infectious disease. It threatens about 3 billion people in 94 countries of the world. Malaria mainly rages in sub-Saharan Africa, but also in Southeast Asia and Latin America. In 2013, more than 200 million cases of malaria and more than 600,000 deaths have been reported. Particularly children under the age of 5 and pregnant women are affected. This high number of malaria victims represents one of the largest tragedies of our century. The situation is worsened by drug resistance of the parasites, by a lack of medical aid, and by political factors. There is still no efficient vaccine against malaria. Malaria is caused by the unicellular parasite Plasmodium, which develops in humans and mosquitoes. Malaria parasites are transmitted by the bite of a female, usually pregnant and therefore hungry Anopheles mosquito. The parasites enter the human body and from there they invade the liver cells. Here they multiply and are released into the bloodstream. Next to the parasites, you can see here red blood cells, the so-called erythrocytes, and white blood cells, the leukocytes. The parasites now enter the red blood cells and grow to become so-called ring stages. These develop into young and then into old trophozoites and finally into schizons, which divide into 16 new parasites. The red blood cell ruptures and the young parasites enter new erythrocytes. In the most severe form, the tropical malaria, this happens every 48 hours. A part of the parasites differentiates into male and female gametocytes, which can be taken up by the mosquito with the next blood meal. With this, the developmental cycle of the parasite is complete. 5 factors are required for malaria to be endemic in a region. This is the so-called May, Me, My, Mo, Mu constellation. Malaria parasites, man, minimal temperatures, mud in which Anopheles can breed, and mosquitoes. If one of these factors is missing, malaria is eradicated in the area within a few weeks. The most important clinical symptoms of malaria include strong headache, muscle pain and fatigue, fever bursts and anemia, which are caused by the release of the parasites into the bloodstream, but also neurological symptoms up to coma and death. New drugs against malaria are urgently required. Scientists at the Interdisciplinary Research Center Justus Liebig University Gießen try to improve the situation. Our laboratory focuses on rational drug development. Rational means that you select a specific target molecule, for example, an important metabolic enzyme of the parasite. You then try to inhibit the target molecule with your new drug. To reach this goal, we study the target molecule in great detail. We study its function and its three-dimensional structure. Then we can design specific inhibitors which fit exactly into the binding pocket of the target molecule. Once you have chosen a good target molecule, you can start with the experiments. In the first step, you take the DNA, the material containing the genetic information of the parasites, and amplify the gene encoding your target molecule. This amplification step is done with a PCR, a polymerase chain reaction. After the PCR, the sample is pipetted onto a gel. By applying electric tension, the molecules in the sample can move within the gel and are separated from each other. Finally, you can see the amplified parasite gene as a band in the gel. In a second step, the gene is inserted into specialized bacteria. The bacteria are plated onto agar, the growth medium, and put into a warm incubator overnight. On the next morning, the visible colonies indicate the bacterial growth. The bacteria, which contain our parasite gene, are now placed into a liquid growth medium. Here they again grow and multiply and produce the parasite enzyme, our target molecule, in large quantities. Shaking the medium supports this growth process. In the third step, the bacterial cells are now opened up and the content of the cells is applied to a special material in a glass tube. By this procedure, the parasite enzyme is separated from the other contents of the cell and purified. The quality of the purification can be judged again on a gel. The pure and concentrated enzyme can now be used for further investigations. In enzymatic tests, the parasite protein can now be studied with respect to its function. To achieve this, the purified enzyme is mixed with different substrates and the formation of products is followed on the basis of color reactions. Using this approach, one can investigate which function the enzyme has in the metabolism of the parasite and how important it is. In these experiments, you can also test the first inhibitors against the parasite enzyme. This is a first basis for drug discovery. Sometimes, several millions of compounds are tested against parasites' enzymes in high-throughput screening approaches. The results can be seen on a computer screen. However, for rational drug design, you also need the three-dimensional structure of the parasite enzyme. To obtain this, you first have to crystallize the protein. The purified parasite protein is put onto small glass cover slips. Into the wells of the crystallization plate, a salt solution is applied. The margins of the wells are covered with oil, and the cover slips with the parasite enzyme are put as so-called hanging drops on top of the salt solution. As an alternative to these manual experiments, one can use a pipetting and crystallization robot, which can test several thousand experimental conditions in parallel. In both approaches, the salt solution gradually detracts the water from the drop. Thereby the protein concentration in the drop increases and crystals can form. Some crystals grow very rapidly. Sometimes you can even follow this under the microscope. Other crystals take weeks to grow. Crystals are solid bodies like diamonds or ice crystals, and often they are very beautiful. In a crystal, all molecules are arranged in a strictly geometric way. Like in an apple plantation, they all look into the same direction. Good crystals are therefore clear and have sharp edges. If you expose a protein crystal from different angles to X-ray beams, you can determine the position of the single atoms in the protein and use mathematical computations to solve the three-dimensional structure of the enzyme. It is fascinating to be able to see such a picture of an enzyme. The protein shown here consists of two identical subunits in red and purple, with bound substrates in orange and yellow. If you know how your parasite enzyme, your target molecule, looks like, you can design tailored inhibitors which fit exactly into the binding pockets of the enzyme, thereby blocking its activity. Also, inhibitors which have been identified before can be optimized with the help of the structure. You can also compare the enzymes for malaria parasites in humans and design drugs which only inhibit the parasite enzyme, thus having no or only little side effects. After an inhibitor has been found, it of course needs to be tested on the parasite itself. Its toxicity for humans needs to be excluded, and its efficacy in patients needs to be proven in clinical studies. The rational, structure-based drug discovery has many advantages. However, it is also a complicated and time-consuming process. It is a long way to discover and develop new drugs. Often this process takes more than 10 years. However, for new drugs which are accessible to all people, these efforts cannot be too exhausting. Healthy and happy children in all parts of the world will be the reward.